So I'm Bert Bloom. I'm a plant pathologist with the University of Arkansas Division of Agriculture, and my lab is broadly interested in crop improvement against diseases. And so we do this in a variety of ways. We combine basic research with applied science to try to translate these results into products that benefit growers. We're very interested in resistance to mycotoxins. So mycotoxins are fungal metabolites that are produced during the infection of grains, and these can have toxic effects on livestock and even humans. These compounds are in some ways difficult to study because the production of mycotoxins isn't a constant occurrence. It can be very variable from year to year, location to location, state to state. Even within a field, it can be very variable. So it's a bit of a challenge to study. We're very interested in fumonisins and aflatoxins. Fumonisins are produced by different species of Fusarium that cause ear rots in corn. Aflatoxins are produced primarily by Aspergillus flavus in corn. And aflatoxin is one of the most carcinogenic, naturally occurring chemicals known to mankind. And so, of course, its presence in grain is of considerable concern. What we're very interested in is trying to develop genetic resistance to these two groups of pathogens with the idea of blocking their ability to produce these toxins. One of the ways in which we're doing this is through a variety of transgenic approaches, where conceptually the idea is to find fungal genes or genes in the pathogens that are involved in either colonization or the production of mycotoxins, and then through a transgenic approach target those genes as the fungus attacks the corn plant. Then the plant is able to turn off those genes in the pathogen. Conceptually, this is what we're trying to do. And so this process involves a lot of steps, and we think of it as a pipeline of discovery. So it starts with understanding the fungal genes that are involved in the regulation of mycotoxin biosynthesis during infection. Once we have those genes identified, we need to make sure that they're actually suitable targets for transgenic approaches. Those genes that pass that vetting process, we then have to build DNA constructs to express a transgene in maize and then go through another series of steps of evaluations to see if it's effective. Yeah, so the ultimate goal of this research is to take these discoveries and take these transgenes and move them into commercially available hybrids, high-performing hybrids that would be effective in the field.
Hi, my name's John Ridenauer, and I'm a PhD student here at the University of Arkansas. I work for Dr. Bert Bloom, and I study a fungal pathogen of corn called Fusarium. And I'm interested in how this fungus is able to infect corn and produce mycotoxins. And one of the tools that we use to identify genes that are involved in infection and toxin production is a reference genome. The genome for Fusarium has been fully sequenced, and we can turn to this to identify potential targets. Once we've identified potential targets, we can use functional genetics in the pathogen. We've identified a gene that's only present in certain fungi, and we found that this gene is involved in both infection as well as toxin production. And one thing that we find very interesting about this specific gene is that it seems to be limited to fungi. So we don't find similar genes in humans or in plants. So we believe that this could represent a potential target for fungicide development and mitigating some of the health concerns associated with fungicide applications.
Hi, I'm Jonathan Smith. I'm from Northwest Arkansas, and I work in Dr. Bert Bloom's lab on methods for controlling aflatoxin. Aflatoxin is a toxin produced by Aspergillus species when they infect different crops, including corn. And it's extremely carcinogenic, and one of the things we're doing to try to eliminate toxin accumulation in corn is developing transgenic maize lines that silence genes in the fungus. Silencing is a way of reducing the amount of gene expression for a specific gene in an organism. In our case, what we're doing is expressing a construct in the plant that will silence specific genes in the fungus. Reduction of the expression of these genes will in turn reduce the amount of aflatoxin produced by Aspergillus. The ultimate goal of producing lines that can be grown and are essentially resistant to aflatoxin accumulation.